Hello students, this is Dr. Ravi Saini. So today we are going to discuss about the topic that is liver and gallbladder. So basically ये digestive system का बहुत ज़्यादा important part है. Uh, it is usually asked in various exams, whether it is UG exams or PG exams or some sort of other specialty exams. So under the physiological basis, this topic covers the most important part of the digestive system. So that is liver and gallbladder. So basically, liver, it is a dual organ. So basically, it performs both secretory and excretory functions. It helps in both secretion. It helps in secretion as well as excretion of various substances. And it is the largest gland. It weighs around 1.5 kilogram in man. The location is upper and right side of the abdominal cavity, immediately beneath the diaphragm. These are the various lobes that is called bladder. This one is a quadrilateral lobe, right lobe, that is cystic duct, bile duct, that is inferior vena cava, that is portal vein. This one is the caudate lobe. This one is the left lobe. So this is the functional, very basic anatomy of the liver that is required in physiology. Now come to the functional anatomy. So for functional anatomy, basically these broad lobes which we discussed in this image, these are the various lobes. So these lobes could also be divided into small functional units. These are called hepatic lobes. So these are the hexagonal lobes. This whole liver could be divided into these hexagonal lobes, which are called hepatic lobules. And in this image, that is representing one hepatic lobule. So each lobe consists of many lobules, and these lobules are called hepatic lobules. Now come to the structure of hepatic lobule. So it is a structural and functional unit of liver. So around 50,000 to 1 lakh lobules in the liver. It has a hexagonal or honeycomb-like structure that is made up of various hepatocytes. These are the liver cells. Now come to the hepatocytes or hepatic plates. So hepatocytes are the liver cells. Basically, we divided liver into various lobes, right lobe, left lobe, caudate and quadrant lobes. And these lobes further divided into hepatic lobules, which are the hexagonal structures. And further, these hepatic lobules are made up of hepatocytes or liver cells. These cells are basically arranged in columns, which forms hepatic plates. So each plate is made up of two columns of cells. In between these two columns, a bile canalicus is present. These are the hepatocytes. So basically, one hepatic plate is formed by the column of these two columns. These two columns, which are formed by the hepatocytes, and there's a space between these columns. This space is called bile canalicus. This whole structure is called hepatic plates. These are the two columns formed by the hepatocytes, and between these two columns of hepatocytes there is a space which is called bile canalicus. And these two hepatic plates, between these two hepatic plates there is a space that is called sinusoid, and which further opens into the central vein, and finally it leads to the hepatic vein. So in between two neighboring plates, a blood space is present that is called sinusoid. This space is called this space is called sinusoid. This space is called bile canalicus. These are the two hepatic plates. These are the two hepatic plates. Between these two hepatic plates, there is a space called sinusoid, which opens into the central vein, and finally it leads to the hepatic vein. And one hepatic plate formed by two columns of hepatocytes. Sinusoid is lined by endothelial cells. In between these endothelial cells, some special macrophages like Kupffer cells are present. So whole liver was divided into hepatic lobules. We studied about one structure of hepatic lobule. This one is a central vein, bile canalicus, that is interlobular hepatic vein, interlobular hepatic artery. These are the sinusoids, Kupffer cells. So between these two columns, this one is the first column, this one is the second column. So between these two columns of hepatocytes, there is a space present which is called these two hepatic plates. There is a space which is called sinusoid, and it opens into the central vein. These are the hepatocytes. It is we could have a very a more clear view in three-dimensional form. These are the sinusoids which opens into the central vein. This is the central vein, which is finally leading to the hepatic vein. So these are the sinusoids which are leading to the central vein. And finally, central vein is leading to the hepatic vein. Between these two columns of hepatocytes, bile canalicus is also present, which opens into bile duct. And finally, it will form hepatic duct, a common hepatic duct. After exit from the liver, it is called extra hepatic duct or common hepatic duct, and finally it is connected to the cystic duct, which is coming from the gallbladder. And finally, there will be some more connections we'll discuss in further classes. Now come to the portal tribe. So these are the hepatic lobules. These are two hepatic lobules. So at the corners of these hepatic lobules, these are specific structures which are called portal triad. Basically, it consists hepatic artery, portal vein, and bile duct. So these three structures are present in the form of triad, and these triads are present at the corners of these hexagonal structures, which we are calling we are calling them hepatic lobules. So branches of hepatic artery and portal vein opens into the sinusoid. Sinusoid further opens into the central vein, which finally opens into the hepatic vein. So hepatic artery and portal vein opens into the sinusoid. Sinusoid opens into the central vein, and finally it is opening into the hepatic vein. And sinusoid is the structure which is present between two hepatic plates. These are two hepatic plates. This is the first hepatic plate, second hepatic plate. This space is called sinusoid, and these hepatic plates are formed by the two columns of hepatocytes. Between these two columns, there is a space which is called bile canaliculus. This space is called bile canaliculus. This one is the column of hepatocytes, which forms hepatic plates. These are the sinusoids, which opens into the central vein, which finally leads to the hepatic vein. And between these two columns, bile canaliculus is also present, which is finally opens into the bile duct. Again, we are repeating the same thing. These are the sinusoids, which is finally opening into the central vein, leading to the hepatic vein. Now come to the portal triads. So basically, bile is secreted by the hepatic cells, which opens into the canaliculus. From canaliculus, the bile further enters into the bile duct. So these tributaries of bile duct from canalicular finally forming small bile ducts. These small bile ducts joins together and forms left and right hepatic duct, which emerges out of the liver and finally forms the extra hepatic duct. It is an extra hepatic duct. Right and left bile ducts, when coming out of the liver, forms a common hepatic duct. which further unites with the cystic duct. So here is the this is the gallbladder. This is the cystic duct. These are the right and left hepatic duct. It is emerging out of the liver and forming common hepatic duct. This common hepatic duct, after joining with the cystic duct, is forming common bile duct. Here, pancreatic duct is coming, and finally, it joins here and opens into the duodenum by the ampulla of the bladder. So there are sphincters present, which is called sphincter of Oddi or pancreatic sphincter, which guards the opening of these two respective ducts. Sphincter of Oddi is responsible for the opening of common bile duct, and pancreatic sphincter is responsible for the opening of pancreatic duct. And finally, these two ducts, after joining through the ampulla of Vater, opens into the duodenum. So initially, bile was secreted by the hepatic cells and enters into the canaliculus. These canaliculus forms tributaries of bile ducts, which leads into the small bile ducts. These small bile ducts forms left and right hepatic duct, and after emerging from the liver, these right and left hepatic duct forms the common hepatic duct, which opens with the cystic duct and forms common bile duct, which is further connected to the pancreatic duct and opens into the duodenum by the ampulla of the Vater. This whole system basically is called biliary system. This whole system is called biliary system. Now come to the sphincter of Oddi. So basically, sphincter basically it is a valve which controls opening or closing, which is present at the lower part of the common bile duct before it joins the pancreatic duct. So basically, this one is the liver. Here, a gallbladder. This one is a cystic duct. Right and left, hepatic duct, common bile duct. This one is a common bile duct. Cystic duct, common hepatic duct, right or this one is the right or left hepatic duct, common hepatic duct, cystic duct, and this one is a common bile duct. So at this portion, this sphincter of Oddi is present. Before it joins, this one is a pancreatic duct. So here there will be a sphincter for pancreatic duct. This one is a sphincter of Oddi, and finally it leads into the this one is a duodenum portion of small intestine. Here we have an opening of ampulla of Vater. So generally it is kept closed. So the bile secreted from the liver cells or the liver enters the gallbladder where it is stored. So usually gallbladder has a function of storage of bile. Upon appropriate stimulation, sphincter opens and allows flow of bile from the gallbladder into the intestine. This is a more detailed diagram of these these sphincters, mainly sphincter of Oddi. This one is a common bile duct. Common. This one is the right hepatic duct, left hepatic duct, This one is the cystic duct, and finally it is forming common bile duct. So common bile duct. Finally, here is the sphincter of hepatopancreatic, and this one is there's a sphincter of pancreatic duct. This one is the sphincter of common bile duct. This one is the ampulla of Vater, which is opening into the duodenum. Sphincter of common bile duct, sphincter of pancreatic duct. This one is the ampulla of Vater, or here we could call that sphincter of Oddi. Now come to the enterohepatic circulation. So basically, there is a portal vein that is formed by the superior mesenteric vein and splenic vein. It brings deoxygenated blood from stomach, intestine, spleen, and pancreas. This deoxygenated blood is carried from intestine, from stomach, from spleen to the liver through portal vein. This portal vein opens into the sinusoid. These are the sinusoid which we studied in the hepatic lobules. These sinusoids open into the central vein, and finally central vein will open into the hepatic vein, and hepatic vein opens into the inferior vena cava. This one is the another diagram of hepatic portal system or enterohepatic circulation. Now come to the what is the importance of that portal circulation or enterohepatic circulation. Basically, the portal blood. Portal blood means the blood which is carried from intestine, from stomach or spleen. This deoxygenated blood is carrying towards the liver by the portal vein. So this the blood which is present in portal vein carries or rich in monosaccharides, amino acids. It also contains bile salts, bilirubins, various GI hormones. So these are very important organic compounds which need to be reabsorbed before excreting into the excreting through the intestine. So this reabsorption through intestine, stomach, or spleen is done by the portal vein, and this is finally called enterohepatic circulation. Hepatic artery basically mixes its blood from the portal vein in sinusoids, and these sinusoids open into the central vein, and this central vein finally opens into the hepatic vein, which leads deoxygenated blood into the inferior vena cava. Here we having portal vein finally opening into the various sinusoids, and finally here it comes as a hepatic vein and opens into the interior vena cava. Now come to the secretion of bile. Basically, bile is secreted by the hepatocytes, the liver cells. Initially, bile is secreted by the hepatocytes, which contains bile acids, bile pigments, cholesterol, lecithin, and fatty acids. So from these hepatocytes, bile is released into the canalically. From here, it passes into the small ducts and finally opening into the right and left hepatic duct, which finally opens into the common hepatic duct, and this common hepatic duct forms common bile duct after joining with the cystic duct, and finally this common bile duct reunites this common bile duct unites with the pancreatic duct to form the common hepatopancreatic duct, which finally opens into the duodenum through the ampulla of the Vater. There are sphincters, specific sphincters, sphincter of Oddi, sphincter of Oddi, and sphincter of pancreatic duct. Sphincter of pancreatic duct. These two sphincters control the movement of the gall bladder. There is a sphincter of Oddi. There is a sphincter of pancreatic duct. This is the ampulla of the Vater, common bile duct, pancreatic duct, cystic duct, right and left, hepatic duct, common hepatic duct. Most of the bile from liver enters the gall bladder, where it is stored. So basically, gallbladder is having a function of storage. From the gallbladder, bile is released into the intestine whenever it is required. Whenever there will be a stimulus in various forms, either presence of food or various under the influence of various hormones, this bile is released into the intestine. So when bile is stored in the gallbladder, there are some specific changes which are related to the quality and quantity. The first one is the volume is decreased because of the absorption of large amount of water and electrolytes. So the first one is there will be a decrease in volume. Concentration of bile would be changed. Bile pigments, cholesterol, fatty acids, and lecithin will be responsible for increased concentration. pH would be decreased. Mucus would be added into the bile. So these are some qualitative or quantitative changes which are occurring in the bile when it is stored in the gall bladder after secretion from the liver.